Hello, everyone. So the study I'm going to talk about today, it's still ongoing, and it's an area that I'm just moving to. So I'm very excited to have the chance to share the overview of our project and some preliminary results. So we're all aware that the drought event is becoming more and more frequent, and it caused obviously damage to the forest, and in some countries it has a severe drought damage or mortality. So us as a forest geneticist should take the responsibility trying to work on improve the drought resistance. And many of the study has been working on it. So I'm also a forest geneticist, but I'm going to talk about the phenotype part because I don't know, at least what I feels that the development for the genotyping and phenotyping is very imbalanced. So we have here so many studies in the past 2 days, also some talks today, so much development in the genotyping, and there are high throughput genotype data available, but for the phenotype part is far left behind. So we're still using the traditional method, which often has it works well, but it has its limit. It's often time-consuming, labor-intensive, and requires destructive sampling. So all these lead to that we cannot have a huge amount of phenotype data. So compared with genotype data, we are lacking the link. Luckily, with the development of the remote sensing and also the platform of big drones can carry different sensors fly over the big forest area, so provide a big remote sensing data, and from those data, there's many information features can be extracted, which link to the phenotype traits we are interested in. So this method, compared with traditional method, is fast and efficient, low cost, non-destructive, and allow us to have a large-scale data. And most importantly, is to be able to measure some traits, which is not easy to measure normally, manually. We cannot even see by naked eye, so we need to use some equipment. So this method has a great potential, trying to find the link between the genotype and the phenotype. Of course, also have the hope to advance the tree improvement process. So with this background, I'm going to talk about the drone drought project. It's a small project, so it's mainly trying to evaluate the potential of this method. So there's two parts of this project. One is in greenhouse, so on small potted trees, so very controlled study, and another part is in the field, forest plantation. But we still artificially create drought by put plastic cover over some selected plots. So in both studies, we used the drone method and the traditional method to do the phenotyping. And so we get different traits related to the drought stress. And we are hoping to find a correlation between the two methods. And the reason why we have a greenhouse control study and in the plantation is because we want to validate the results we can find in the controlled study can also observed in the natural stand. So in this project, we have Katrina, who is an expert in remote sensing, and Ole, and me as a forest geneticist, and Anas, who is a physiologist. So we have almost all the experts needed in this project, which all in-house in our department. Oh yeah, forgot to mention. So the trees we have been using is Abies nottemiana. Well, it's not only because it's an important Christmas tree species, but it's because we have the clones available. It's a somatic embryogenesis clones. So we use clones trying to eliminate the genetic variation among trees, so only to focus on the phenotype method development. First part, for the controlled drought treatment, as you can see, this is the whole area for our study. It's very colorful. So there are 3 different colors indicate 3 treatment. Blue for the control, yeah, and I will not talk about time. So we have 32 trees from 5 clones in each treatment, in total 96. And how do we just to mention that we use some silicon pens to cover the potted trees to avoid the rain affect the treatment. We want to do it outdoor because we want to have the natural light to make it as close as possible to do like in the plantation. So when we see the 3 treatment, how do we evaluate they are different? So we use the pre-dawn water potential as the variables to quantify this. So this is a reliable indicator of plant water status, but it requires measuring before the sunrise. And we had this experiment in summer. Well, if you have been in Nordic countries, you know Denmark never really get dark, so sunrise. Well, so luckily we figured out this method that we also luckily using the potted trees, which we possible to move the trees into our basement at our institute and cover with plastic to simulate the darkness. So I think that's brilliant, but rough method. Then we can able to measure the water potential in the next early morning day, so we don't have to wake up 2 o'clock. Yeah. So the water potential showing that, so at the beginning, all the trees was watered to the full capacity, so they have the same water potential. So then we continuous water for the control trees, but stop watering for the dry treatment. And for the intermediate, like the medium dry, we water less frequently. After 22 days, you can see that the dramatic drop for the water potential for the dry treatment. This reaching to the, I hope I can say this, is a permanent wilt point. The physiologists know that. So they cannot go lower, so then the trees will die. So then we try to keep at this water potential level by monitoring the pot weight or soil water content. I don't want to measure this water potential all the time because it requires lots of work. And so later on, we just make it simple, use soil water content to make sure they are constant. And it also based on results showing they are more or less. So what we can see from this is how the trees looks like from the 3 treatment. So the dry ones apparently has the symptom, but the other 2 are hard to see the difference. That's again see that naked eyes cannot see everything. So we need the machines. So what we need to do now, we need to find the correlations. So for the remote sensing part, because the small trees and it's in the campus area, so we couldn't use drone. Instead, and it's a small project, we don't have the budget to use the fancy high throughput study has previously presented. So instead, we again made some inventions. We created this platform similar like the high throughput phenotype lab. So instead of automated move the trees, we manually move the trees to under each sensor. It's doing same work, but with a much lower cost. So, and the sensors we have using thermal camera, multispectral camera, and the gas analyzer. And then the 3 traditional used equipment, which normally used to measure the water stress. Previous study presentations already talked about this. I will not go further. Just to show you the table, that's the vegetation indices we have been used so far in our study, try to find the correlations. So we only have 5 bands for the multispectral camera, but it's already allow us to use so many vegetation indices. Gas analyzer we used to measure the CO2 and the water fluxes. There's a formula and explanation given by the remote sensor expert, which hard to explain for me, but if you are the expert, just give you the information. So as I said, after 22 days, there's 3 treatments showing obvious difference, and we keep at a constant level. Then, since then, we start to measure using 2 different methods repeatedly for 6 times. And today, I'm only going to talk about one of the measurements in the middle, which is 2 weeks after the drought has been created, as a most stable and extreme case. So this is, obviously we just want to give a first quick question. Is there correlations between these 2 different methods? So what we really interested in to look at is this part. So that's from the traditional method. There's much more trees we measured, but we already tested they are highly correlated. So in order to give a smaller table, I removed the majority of them, only keep the transpiration and the soil water content. And all the non-significant correlations is a black cell. So if you see the cell with the color and the numbers, they are all significant correlations. So those correlations are linked to those vegetation indices from the multispectral camera, and also from the water flux from the gas analyzer. So this has a significant positive correlation. So it means if there's a higher soil water content, it means there's a higher water flux, which makes sense. So, but again, in order to get a similar results, use the traditional method takes a very long time, but using this gas analyzer, it was super fast. I used 3 days to finish the traditional measurement for 96 trees, and Katrina only used 3 hours to finish. So that's a difference I want to see. This is the summary table for the vegetation indices, which has a high significant correlation with the drought stress traits. So now we want to see, can we see the same results when we go to the forest stand? That's also one question from previous presentation. So we have this Apis nodminiana clonal trial was established in 2014. So we are allowed to do this experiment to cover some of the plots with plastics. So we covered the last September, so until now has 1 year. In total, there's 87 trees are inside the plastic cover. So these are the dry treatment, and the 79 trees are outside the plastic cover as a control. Then we repeat the same method. The only difference, we include the laser scanner in the field, because those are big trees. It makes more sense to use them. And small trees, it's not worth to do it. And also, yeah. And in the field, now we only possible to measure the water potential and the photosynthesis yield because the other equipment needs the constant power supplies and constant light, so which is not possible in the field. Now, in the field, we cannot move trees to basement anymore, so we have to move ourselves to the field at night. So we had this ground measurement in July before the summer holiday. That was a nice relief. Luckily, have some students help me with that. And so I wasn't sure if the plastic cover really helps to create a drought, but the results from the water potential showing here, there is significant difference between the trees from these 2 treatments, which is great. So there has some maps created by the different sensors, which before the drought treatment, and also the LiDAR for the height. And we just collected the data for after the drought for the remote sensing part. So unfortunately, today I don't have results to show. So it will come, so we will see if they also showing the same results. Also, I don't have time for that, so next time. So the conclusion so far, there's correlations from some sensors, which is great news, and which will hopefully in the future can be more developed and validated, can be routinely used in the tree improvement program. So lastly, that's people working in my project and the founders. Thank you so much for your attention, and I would highly appreciate it for some comments and discussions, because it's still ongoing, so thank you.
Thank you very, very much for the nice talk. We have time for a few questions. I can ask the first one. Probably you said it, but I missed it. What's the final goal to use the drones for these plantations? Is it to generally check if more watering is needed, or is it to see the differences among the different genotypes?
Yes, it's like to develop an alternative phenotype method. So we want to eventually to use as a phenotype method to see the variations of what we observe, like the water stress level. Of course, we'll link to the genetic. So yeah.
So you're interested in the differences between the genotypes, yeah?
Yes.
Okay.
Mm-hmm.
Thank you.
Okay, that is not mine.
Jan?
Why did you decide to put the shelters or cover the plots or not the individual trees, randomized through the site? Because it's more convenient, or?
Well, yes, no. I mean, yes and no. So the thing to cover like the trees in the neighboring stands, right? So they also, if you cover individual trees, they still has a higher chance they can get the underwater from the neighbors. So if you cover big area in the middle of the plot, plastic cover, there will be more drought treat. Yeah, just to create an extreme drought case.
Yeah. Thanks.
Yeah.
Thanks.